In this poster, we present ConAn, a tool in R package built for the differential analysis of co-expression network connectivity. In omics data, co-expression networks are sets of genes that display correlated expression levels across samples and are frequently functionally related, such as sets of genes in a biological pathway. A set of co-expressed genes is called a gene module, as seen here on the right, and can be represented in a network graph where genes are shown as nodes and the dependency between them are represented by edges, and is termed connectivity. The connectivity of a module has nothing to do with gene expression levels, but rather the degree of co-regulation of the genes. Examining the differential connectivity of a module between two conditions, such as healthy and disease states, can identify aggregate changes in the rewiring of connections associated with disease, which may be missed when looking at changes in individual genes, such as through a standard differential gene analysis. On the left, you can see the standard pipeline for running ConAn. It takes as input an omics data set and a set of predefined gene modules associated with this data set. Any type of program can be used, such as WGCNA. ConAn assigns each module a module differential connectivity score, or MDC, which corresponds to the change in connectivity of a module between conditions. An MDC score above 0 indicates an increasing correlation between the genes within a module. After each module is assigned an MDC, permutation testing is performed to determine a p-value for each MDC. For a number of iterations, the condition labels for each sample are permuted, and the MDC is recalculated, resulting in a distribution of null MDC scores, as seen on the right. The orange line represents a true MDC score that lies within the null distribution and would not be significant. On the right, the green dotted line represents an MDC score that lies outside of the null distribution and would be significant.
Simulation studies were performed to test the sensitivity and specificity of ConAn through power and family-wise error rate evaluations. A simulated set of differential networks were made to test ConAn's ability to detect differences in modules with varying levels of initial connectivity in one condition, as shown on the x-axis, with varying levels of changes in connectivity between conditions on the left y-axis, and varying levels of background connectivity on the right y-axis. The power evaluation shows that as the difference in module connectivity between two groups becomes larger, ConAn's discovery rate is increased, as shown in red. The family-wise error rate evaluation shows that ConAn can achieve very low error rates across all levels of variation when background correction is applied, shown by the red line.
Finally, ConAn was applied to published expression data of breast cancer samples from the Cancer Genome Atlas. We compared module connectivity between two molecular subtypes of breast cancer, basal and luminal A. And as you can see, one module was much more highly connected in the luminal A group than in the comparative basal group. And you can sort of see the null distribution on the right-hand side. And you can see that the true value, MDC value, lies towards one extreme of the distribution. If we go on to the next slide, we'll see that a visualization of the module. This module has as a hub gene PLIN1, which has been previously implicated in breast cancer mortality in both estrogen receptor positive and luminal A breast cancer subtypes. In conclusion, we've presented ConAn, a tool for differential network connectivity analysis of expression data. ConAn can effectively identify changes in gene modules across conditions, as shown through rigorous simulations and a recapitulation of published characteristics of breast cancer. And we'd also like to thank all the people who have helped us out during this process: Dr. Stefano Monti, Eric Reed, Anthony Federico and all the other Monty Lab members, in addition to Dr. Gary Benson, who provided a lot of feedback to us during this project. Thank you.